All right. Well, I think it's a good time to maybe get started here. So I just want to thank everybody again for joining us today. My name is Norman Ong, and I'm one of the co-founders here at Constantium, alongside Nicholas Schaefer and Daniel Goleman. Today we have the pleasure of having Dr. Victoria Parikh, who's a clinician scientist who cares for patients and studies genetic cardiovascular disease. And she'll be diving into how cutting-edge high-throughput functional assays, typically called multiplex assays of variant effect, or MACE, are revolutionizing the way we approach genetic variant interpretation, offering new insights into cardiac conditions. So Dr. Parikh is currently the director of the Stanford Center for Inherited Cardiovascular Disease, which is one of the largest of its kind in the country. So Dr. Parikh is also Constantium Biosciences clinical advisors, and we are very lucky to have other advisors who are part of our advisor board, and obviously are very valuable in providing us with a lot of insightful advice to develop and commercialize evidence, which is our core offering, our premier functional evidence for clinical variant interpretation. Today, you also hear from our co-founder and CEO, Nicholas Schaefer, who will provide an overview and demo of the evidence at the end of today's event. As you probably already see, today's session will be recorded, so we'll send out recording after the session. And if you have any questions at all during the webinar, feel free to enter them under the Q&A section, and we'll respond to them at the very end of the session. So without any further ado, I'm going to hand it over to Dr. Parikh. So, enjoy the session, and I look forward to a productive event.
Thank you so much, Norman. It's really a pleasure to be here, and thank you everyone for taking the time. So we are working together, Constantium and I, to try to help to bring awareness today around the role for MAVs, or multiplex assays of variant effect, and the data that they generate in cardiovascular medicine. So I think the first thing to do, because in cardiovascular medicine, the role of genetics is, I think, a little bit less clear, especially Mendelian genetics, which is what many MAVs study. And so I wanted to just talk about how actually Mendelian variants cause a significant minority of cardiovascular diseases. What I've done on this table is to list the diseases that we classically think of as being caused in the heart by genetics. So familial hypercholesterolemia, which of course causes early heart attacks and strokes, and very high LDL, or bad cholesterol, has a very high prevalence that we didn't really recognize in the population, estimated in as many as 1 in 200 individuals. And then, of course, recently, hypertrophic cardiomyopathy, which we now know to be very much a Mendelian genetic disease, we initially thought to be hyper rare, but as we have actually developed more and more precision therapies, thanks to the hard work of Cricket Seidman and Leslie Leinwand, looking at the actual mechanism of disease, we've actually uncovered many, many more cases. And so the estimates are actually much higher than we thought, 1 in 300 to 1 in 600 individuals with hypertrophic cardiomyopathy. And then as we move down the list, it becomes slightly more rare to see these syndromes. But as you can see, when we add them all together, we really actually get a significant minority of patients with cardiovascular disease who will have one or more of these Mendelian variants. And we know this is true because when we look in cohorts, as you can see here on the right, of cardiovascular disease and general cardiology clinics that offer genetic testing, we find that there are a significant percentage of positive tests. Most of these, of course, as you might imagine, explain dyslipidemia. But a significant proportion are cardiomyopathy and other genetic diseases. And then even when we step back and look in biobanks, which are the general population, so to speak, we find variants that are causative. So this figure is actually from a really great paper out of the UPenn Biobank led by Joe Park, showing that if you look for truncating variants in lamin AC, which is a gene that causes a plethora of different diseases, including a dilated and arrhythmogenic cardiomyopathy, but also progeria. If you look at those patients in that biobank that carry lamin, they have significant associations in their phenome with parts of those diseases that we know to be caused by Mendelian variants. And so not only are these variants common in a known disease population, but they also cause disease in a population that we think of as otherwise not carrying rare disease. So it's really important in cardiovascular medicine to be able to identify patients with these genetic diseases so that we can then develop precision therapies for them. So as you know, variant interpretation to be able to find folks who carry damaging variants, causative variants in these genes of interest has multiple different types of evidence that can support it, but often come up short. So for example, we can use the population to look for the rarity of these variants, but we know that whether or not variants are causative of disease, they tend to be rare. And so using rarity alone very, very rarely actually allows us to assign pathogenicity. It's actually more useful for ruling out benignity or more common variants as monogenic causes of disease. And we don't actually, see all of the variation in the entire population that may be causative of disease. So being unable then to do a preemptive analysis with the population of new variants that come into the clinic is also extremely limiting. Then, of course, there's family segregation, which is extremely slow, and especially in the US, very hard to get a hold of family members who actually have disease, adequately phenotype them, and identify their variant of interest. Computational predictions, although they are improving with, for example, alpha missense and other protein structure models and splice AI, are still lacking in their ability to actually decide whether or not a variant can change the course of human disease in and of itself. So then what we're left with is functional data. And arguably, this is one of the strongest contributors to our understanding of whether or not a variant can actually cause disease, because it tells us whether a variant can actually disrupt the gene protein's function. But I think that we are just very slow at this, right? Because until now, we haven't had a way to sort of preemptively or prospectively look at every variant in genes of interest. And so what we end up with is very slow functional characterization of single variants in single families, which then are rarely applicable to more than one family or patient. So as we've talked about, the vast majority of genetic variants then that we find on genetic tests end up being variants of uncertain significance, or VUSs. Even in the best studied genes in the best studied diseases, for example, BRCA1 in cancer, 88% of the variants that are possible in that gene are still variants of uncertain significance. So what this means is that the VUS problem is severely hindering our ability to manage these patients medically. So when you see a patient, when we think about sort of the how big this problem is, I think that we end up looking at multiple sources, and what you see is that across cancer, across cardiovascular disease, and across really professions, including of course genetic counseling, we find that VUSs actually are creating a significant burden of uncertainty for patients and for medical professionals. So in cardiovascular disease, the natural history looks like this. Patients inherit a causative variant. There's either they develop symptoms or they don't. And then if they develop disease from that variant, they can end up with sudden cardiac death or the need for heart transplant. Or early MI, which also can cause sudden cardiac death. So our opportunities for intervening here include genetic diagnosis and disease prevention, which then we hope someday will actually afford us the ability to create precision therapies. But it turns out that because of the VUS problem, we are exceptionally hindered here. As we increase the number of genes that we test here on the x-axis in cardiovascular disease in order to identify the genetic cause of Mendelian disease, we actually just see a rise here in VUS and a drop in benign findings, and then we see a minimal rise in our ability to actually diagnose. So this delta here, this rise in VUS, is really related to the fact that we don't understand what the variants that we identify are doing. So one way that we can address this, as we've been talking about, is by using multiplex assays of variant effect. And the idea here is that we can preemptively establish functional data at scale for variants of uncertain significance in diseases, in genes that cause cardiovascular disease. So what we do in MAVES is we take a cell type of interest. So for us, it's an induced pluripotent stem cell, which we can then turn into a cardiomyocyte. We somehow introduce a library of variants, whether that's with an exogenous construct that can be delivered via lenti or adeno-associated virus, or whether we actually edit the genome in situ, which is how we do it in my laboratory. We then end up with a pool of cells that carries the variants that we're interested in. So we can do this in up to, in hundreds of variants at a time. And at scale, as we do this in parallel, of course, we can get entire genes done. We then can create every possible variant in that gene and then use a cell-based or molecular assay in order to classify those variants as causing a phenotype that looks like disease, or looking at those molecules as being associated with a loss of function in the molecular consequence of that protein. And then we can sequence, right, in order to identify which variants ended up in the pools that were more like the reference genome and which ended up more like the pools that look like they have disease. From that sequencing, then we end up with pathogenic variants, which look, more like they are causing functional disruption. Or we have what we would classify as benign variants, which look much more like the function associated with the wild type genotype. So this is the way that we can enable proactive or preemptive collection of functional data on nearly all the single nucleotide variants in a gene in order to really pump up that functional data, which then allows us to be able to analyze and interpret those variants in the clinic. So we have actually used this in concert with the CardioVar Consortium, which is a multi-center R01, including familiar faces as shown here. For example, Fritz Roth, who is also a founding advisor of Constantium. So this is actually a variant effect map that was created by Dr. Roth and his team. What they did is they took a gene called CALM1, which it has two other homologs in the human genome, 2 and 3. And on the x-axis here, you can see the amino acid position, and on the y-axis is every possible amino acid substitution. So Fritz and his team introduced these variants into yeast, and it turned out that this gene is actually critical for yeast survival. So they could see whether replacing with this amino acid substitution would then allow for yeast survival. Well, it turns out that this gene is important actually in sudden cardiac arrest because it can be associated with two different syndromes, long QT syndrome and a syndrome called catecholaminergic polymorphic ventricular tachycardia, or CPVT. So when we're interpreting variants in CALM 1 through 3 now, we can actually look at this map and we can say, okay, did that particular amino acid substitution not change the ability to rescue yeast survival, or did it in fact look like it wasn't adequate to replace the native gene in yeast? And what you can see here is that specific variants of uncertain significance actually are highlighted with different levels of likelihood of pathogenicity. So I'm going to show you now how we have been able to apply this in the clinic with 3 cases. The first case was a patient who, this is a pediatric patient who presented with palpitations, actually had quite a long QT, as you can see here on their baseline ECG, and then on exercise actually ended up having significant biventricular, or rather bidirectional bigeminy. And so This patient was sequenced and 2 variants were found, both actually in CALM-2. Neither of them was present in population data sets, and there were no prior case reports to be able to clarify the pathogenicity of these variants. And the parental genotype, which is important in these cases because it can tell us whether the variant is de novo, which is a point toward pathogenicity, was unknown. Then for case 2, it was a patient who fell off the monkey bars and syncopized and was found to have an extremely long QT associated with long QT syndrome, which can be from CALM genes. In CALM-3, another missense variant was identified, again, not present in the population or in case reports. This parental genotype did show that the variant was de novo, which would push it up to pathogenic status in our current system of interpretation. And then in case 3, we actually have a 17-year-old who died in his sleep, unfortunately, and had a really strong family history of cardiomyopathy and sudden cardiac death. But it turned out had a variant in CALM-1, which has not previously been associated with cardiomyopathy. The patient's autopsy did show some fibrosis in the myocardium, and it turned out that the variant was de novo. And so the question was, is this variant pathogenic? It looks like potentially it was contributory, but was it the entire cause of this family's disease? So we went back to the variant effect map here to improve our genetic diagnosis for these patients. And what we found was that for the first 2 patients, the 2 variants that had been identified in that first patient, one looked like it was quite likely to be pathogenic, and the other was somewhat equivocal, but toward the pathogenic side of this graph here. And then the other, which had been found in that second patient that had fallen off the monkey bars, was also in the pathogenic range, confirming that that de novo variant actually did look like it could cause disease in this patient. And then we looked at the third case, which was the most confusing because the variant was de novo, but the phenotype in the family didn't seem to fit CALM disease alone based on the cardiac fibrosis and family history of cardiomyopathy. And it turns out that this variant actually did not have a high likelihood of changing the function of the CALM gene. And so we think was less likely the sole cause of this patient's disease and led us to continue to screen his family for cardiomyopathy and other causes of disease, rather than saying that this de novo variant was the entire cause of his sudden cardiac arrest and his family members no longer needed to be screened. So we have extended this type of work. These are 2 really beautiful papers from our colleagues, Dan Roden, Andrew Glazer, and Brett Cronky at Vanderbilt, who are also part of the CardioVar Consortium, looking at other causes of long QT syndrome. This is KCNH2 here on the left, which is the potassium channel, and this is looking at the pore of the potassium channel with single nucleotide substitutions as seen here. And then additionally over here is showing that when you do the same in a different gene, which is the sodium channel, SCN5A, you actually have a great deal of ability using ACMG criteria and using the MAVE data as functional evidence in reclassifying VUSs, with 35 of the 61 VUSs that were found in patients reclassified to likely pathogenic. So with that, I'm going to turn it over to the Constantium team to tell us a little bit about how Mavidence actually takes that functional data we were looking at and makes it useful for providers and scientists in understanding the cause of genetic cardiovascular disease.
Great, thanks. Yeah, as I mentioned, thank you, Dr. Parikh, and thanks to everybody for joining us today. My name's Nick Schaefer, and as was mentioned, I'm one of the co-founders of Constantium Biosciences. So several years ago, my co-founders and I set out on a mission to make it possible for everyone involved in clinical variant interpretation to easily and responsibly leverage MAVE data in their workflows. And at the time, the key trends we were noticing were the rise in VUS as a result of more patients having larger panels, like Dr. Parikh mentioned, alongside the rise in the availability of MAVE data that could, at least in theory, be used to help resolve those VUS. And we talked to stakeholders throughout the industry and found that variant scientists both overwhelmingly wanted to leverage MAVE data to improve interpretations for patients, while at the same time they were overwhelmed by the challenge of trying to keep up with the release of data and the analysis of the data. And so these conversations inspired us to create Mavidence, which transforms the painful process of trying to find and sort through these complex data sets into a simple search saving 90% of the time that it would otherwise take to leverage MAVE data, while also increasing the VOS resolution by 35% where the data is available based on 2 large-scale studies that we've performed with trial users of Mavenets. So I'm going to jump into the application here in a minute and show you around. But please know that you can have a look around yourself by following the link here that's on the screen and clicking sign up to create an account. Great. So our landing page here, in addition to having links to the user guide and the API documentation, it's got 2 different ways that you can get started looking through the available data. The first is the variant search page, where you can enter your gene of interest as well as your variant of interest and click Get Variant Report. On this page, there's also shown at the gene level the overview of the available coverage of the gene from the various assays that are in the system. There's also the variant overview page. And this page gives you this access to the same data, but in a different format. In this case, you can choose the gene that you're interested in, let's say COLM1. So I'll type in COLM1. This is one of the genes that Dr. Parikh highlighted in her portion of the presentation. And then you have access to available filters and sorting so that you can find the data that you're looking for quickly. I'm going to go ahead and type in a search for one of the variants that we saw highlighted in Dr. Parikh's presentation. And so from here, once you find the variant that you're interested in, whether you're on the variant search page or the variant overview page, clicking the View Report button will take you to a report that summarizes the evidence for that particular variant. So in this case, we're looking at the report for D94A inside of COLM1, and each report contains at the top a summary of the available evidence, including an overall recommended ACMG code, a summary of the relevant gene functions for the gene that we're looking at, as well as a series of evidence panels. And there will be one evidence panel for every piece of data that's available for this variant. In this case, there's only one piece of evidence, so we're looking at the one evidence panel. Each evidence panel contains a link out directly to the study where the data was derived from, a summary of some key aspects of the assay in the context block, and then 2 different representations of the odds of pathogenicity for this variant, one in terms of a numerical score, and the other one has been translated into the corresponding ACMG evidence code. As well as those text in each evidence block, we have a graphical representation of the data. And this shows where the score for this particular variant, which is shown as a black drop vertical, is in the assay relative to the pathogenic and benign reference variants that were included in the assay. And so we can say in this case, this particular variant is well within the pathogenic distribution, and it's getting an ACMG evidence code of PS3 moderate as a result. So our goal with Mavenets is to allow you to seamlessly harness the power of Maves and integrate them into your workflows. And we do this by actively curating the literature and analyzing the data as there's new studies appear. As we demonstrated, we provide a variant level search, both through a web application and an API. We do follow those published guidelines where they're available for systematically turning that raw data into evidence so that it can be easily applied, and then provide all of that evidence inside of an easy to follow report, which summarizes the data. If you do work within a larger organization that has its own variant interpretation software, we provide an API integration so that you have the option of not having to go out to a separate application while using Mavenets. And finally, we're here to support you along the way and provide timely support should any issues arise. And at this point, the Mavenets is both a trusted and proven source. So we do have NIH support, where we were identified as one of a small number of companies that are going to have a major clinical impact. We've already demonstrated the ability to rapidly integrate new data sets as they become available. And as I mentioned, based on 2 large-scale studies that we performed with trial customers, we show that we can increase VOS resolution by between 32 and 38%. And based on more than 100 voice of customer interviews that we've done with stakeholders across the field, we believe we can provide a 90% time savings. Okay, great. Well, I think that brings us to the end of our planned presentation. So we're happy to take any questions that you have.
Great. Thank you, Nick and Dr. Parikh, for the great presentation. So yeah, just a quick note. If you guys have any questions, feel free to enter them in the Q&A section. And yeah, so let me see. Maybe we can start off with a few questions. And again, feel free to enter as as we still have quite a bit of time here. So one of the questions that we have here, panelists, is can MAVs be useful for polygenic conditions?
I can start with that and then hand over.
Right.
I think it's such a good question. So the sort of classic definition of a MAVE being that, you're taking a segment of DNA and making every possible variant. So you're sort of carpet bombing that. It's a little bit more difficult, I think, to extend that across the genome for polygenic conditions, but something that folks are thinking about is taking lead SNPs from GWASs and trying to figure out which variants sort of around those areas that those SNPs travel with might actually be causative of disease. The issue is that you need to have a super sensitive phenotype, right? Because you need to make sure that this common variant, which likely has a smaller effect on disease overall, is actually contributing. And if it has a small effect, you need to be able to detect that. So I think thinking about how they change gene expression is probably the right way to do it. And in that case, then you can sort of look at, for example, transcription factor binding sites, et cetera, to identify which common variants might actually be contributing to disease.
Great. Yeah, so I guess the next question that we have is how does MAVE data compare to computational predictors in terms of quality?
I can start and then punt over to you guys as far as what you've seen. So it depends on the computational predictor, as you might imagine. So I think that for Splice AI, for example, we have collaborators who have very early prelim data showing that actually a readout of a splicing reporter for a MAVE of a specific splice site actually really tracks quite well with Splice AI. But I think that for others, we know that that it doesn't correlate as well with what we see in humans. And so we think that actually this is additive and that it may actually be able to sort of be contributory to models that are currently in use.
Very nice. Thank you, Dr. Parikh. Yeah, so another question that we're getting from the audience. So how have you dealt with phenotypic variability within a gene and how it impacts classification of a variant for a specific condition rather than for all conditions attributed to a gene? So the example here is LMNA and arrhythmia, cardiomyopathy, and neuromuscular conditions.
Yeah, thank you for asking that. So I think that you generally want to multiplex your variant assay so that you get something that's very sort of germane to the function of the protein. So for example, if you want to look for, variants that you think are going to be splice variants in lamin because that's going to cause truncation, then, you can do that in a way where you just actually look for sort of RNA or protein expression downstream of making those variants. And then I think that if you're interested in a specific disease, you really want to think about how you model that in a cell type. So for example, for lamin, we can take an iPSC and we can decide whether, does it have trouble differentiating to cardiac muscle or skeletal muscle, right? Does it have trouble differentiating into smooth muscles, which may be part of the problem with progeria overall? Are we looking for senescence, for example, and is that a feature of those downstream diseases? So you're right, you do need some a priori knowledge about the biology and mechanisms that underlie those diseases in order to have disease-specific outcomes. But sometimes you can have an outcome that will affect many, many diseases. And that, I think, comes as you get close closer to the actual function of the protein that the gene encodes.
Thank you. Yeah, so the next question we have is what happens in MAVE when two or more assays don't agree or disagree?
Yeah, so I can take a swing at this, and this is definitely an evolving point. As I mentioned in the presentation about Mavenance, we do try to follow those published guidelines where they're available. And previously, this was almost a non-issue for the reasons that Dr. Parikh mentioned, which is that it was rare overall to have data for a variant, let alone multiple assays for an individual variant. But that's really changing rapidly, and there has been some guidance put out in terms of how to handle situations like this. And so existing guidance suggests that you should find sort of the best overall assay and then use the data from that assay. So that's sort of the generic guidance. Beyond that, there are ClinGen VSEP panels that are working on specific genes and providing more detailed guidance. So for example, within Mavenance for TP53, we've implemented the ClinGen recommendations for combining evidence, and that has more subtle ways of taking multiple assays into account, especially important in cases where you have assays that are sensitive to both loss of function and gain of function types of variants. And so short answer is that there's sort of some generic recommendations that are available, which we follow. There are also on a per gene level, some specific recommendations from ClinGen VSEP panels, which we do try to follow also when they're available. And it will continue to evolve as more data becomes available.
Great, thank you. I think we have time for maybe two more questions. Then I see one more here. So if you have, another question that you are dying to ask, feel free to enter them. So the next one is, do you include splicing information in Mavenance?
So currently, Mavenance is focused on coding variants. And so there's splicing information available within Mavenance in the sense that when an assay is sensitive to splicing variants, that will be mentioned in the assay context. But we don't have a specialized function for identifying potentially disruptive splice variants. And so that's where, in the future, we're looking to combine with things like the types of splicing predictors that Dr. Parikh mentioned to provide a more holistic view on the ways in which different variants can disrupt function.
Great, thank you. Yeah, looks like, yep. One last question, guys. So do you include information on synonymous variants on Mavenance?
Yeah, there are indeed. So, you may have noticed when I was showing the graphical representation in one of the evidence panels, in addition to those pathogenic and benign reference variants, we do show distributions for synonymous and nonsense variants as sort of different points of reference. Those distributions are not explicitly used for the odds of pathogenicity calculation, but especially for assays that have a low number of reference variants, a lot of people would like to see how the assay does in separating those synonymous and nonsense variants in terms of the score. And so we provide that information in the graphical summary.
Great. Yeah, I think it looks like these are all the questions we have for today. I guess, I just want to thank both of you again for the wonderful presentation, and obviously everybody for attending today's webinar. Definitely welcome everybody to sign up for a Mavenance account. You can easily do that by going to mavenance.com. And obviously we are here. If you have any questions, feel free to reach out to us. But yeah, we would definitely keep you posted on any future events of us, and we will be reaching out shortly to share a recording of today's webinar. So yeah, thank you again, everybody, and have a wonderful day and the rest of the week.
Thanks, everybody.
Thank you.
Thank you.